I'm Stephanie Beattie and I'm here to talk about an experiment completed by M.K. Sandberg H, Al-Doujaily B, A, Clarke and J, Collinge. This journal article is about prion propagation and toxicity in vivo and how they occur in two distinct mechanistic phases. Prions are misfolded proteins which cause neurodegenerative diseases. PRP is a cellular protein that is mutated to create prions. Prions gather within the central nervous system of the host. This is where the neurodegeneration occurs. The prions are responsible for bovine spongiform encephalopathy, mad cow disease, also known as Creutzfeldt-Jacobs disease in humans. This disease is where the accumulation of prions cause parts of the brain tissue to become spongy and therefore corrupts the normal brain function. This disease is almost always fatal. This disease occurs in two distinct phases: a clinically silent incubation that is prolonged, followed by a highly aggressive clinical phase. Laboratory inbred mice, wild-type mice with 50% wild-type expression, transgenic mice and TG20 mice were used in the experiment. The mice had expression levels of PRPC, no PRPC, expression of PRPC and 8-fold expression of PRPC. The mice were injected 30 microliters of RML prion inoculum. Mice in groups of 5 or 6 were killed at predetermined time points or at onset of clinical disease. Brains were removed and prepared into cell cultures. The cell cultures were then prepared and sent through the Scrapie cell assay, or Scrapie cell endpoint assay. The experiment's results showed that the TG20 mice with overexpression of PRPC had the shortest survival time, being 59.5 days. The inbred mice with expression levels of PRPC lasted 137 days. And the homozygous mice had the longest survival time of 258 days. However, the inbred mice that had no PRP expression did not develop the disease. Assays determined that by day 10, toxic PRP had fell to undetectable levels. This graph shows that all the mice after inoculation in the first phase, as each mouse type drops from 100%, establishes that the prions have moved from phase 1 into phase 2. The propagation of the prions and their toxicity is shown here. The duplication of the prions is exponential in phase 1 until it reaches the limit. The plateau phase 2 is where it switches to the assembly of toxic PRP. The toxic PRP does not gather until after the toxic threshold and the clinical signs begin to occur. This journal article, published in 2010, is not the most recent study. There is another article in the suppression of prions in the mice that have no PRP expression, that don't have to develop the disease. The understanding shows that the body's natural defences attacks prions and leaves the healthy prions that haven't mutated. Although different experiments, the newer journal articles use results from the article I've just discussed. Thank you.